Okay, so before I start, um, let me just uh, run through what integrative physiology is and what we will be discussing. So the format of the lecture will be primarily based on answering seminar the seminar topic in integrative physiology. So during that topic, we will be, or you will be, going through regulation and adaptation, what the definitions are, and a little bit more information on those topics, and then how they, um, and then regulation and adaptation in the context of exercise, and just the multi system response that occurs for for our body to be able to carry out functions during exercise and demands as well. And then we'll go through thermoregulation. We will also go through reactions to body posture changes. So in that I mean orthostatic reaction, clinostatic reaction, and then to kind of end things, we will go through the physiological reactions and responses to hemorrhage or bleeding. So yeah, so like I mentioned before, we'll try and answer the seminar bullet points. Um, I'm not entirely sure when the second years have their this particular seminar. I think it's coming up for you guys. If it's not, just message me in the comment section, just so I have a rough idea as well. But I think it's an important topic, but I do think it's a very um, simple and logical topic to understand and to kind of grasp and then apply to day-to-day physiology, basically. So we'll go through the physiological mechanisms, um, and then we'll also obviously go through some key definitions, and then like with everything, we'll try and correlate it with some clinical examples. So we have so also we can relate to these topics maybe a bit more easily, and so we can also see um, what they are like in actual practice as well. So yeah, and just another thing. I mean, this lecture it's primarily probably going to be of revision use rather than learning new things, or at least I would expect you to be just reminded of things really rather than learning new things from this lecture. You might learn a few new things here and there, but for the majority I would expect you to kind of have a basic grasp of it, and this will just hopefully iron a few things out or simplify things. So yeah, yeah. So I like to start off with study tips and final exam advice just before we start everything. So when I was studying physiology, I used to revise using active recall. So what active recall is, essentially you go through a topic and then you actively recall it. So you possibly test yourself. I know that there are a lot of um, apps and platforms which you can create your own flashcards and revision notes, and then you can test yourself after reading the particular topic or a particular abstract, for example, as well. And especially for physiology, I think active recall is very, very important because it's very, very systematic. It's almost step by step by step. and so you kind of need to drill yourself into having some nice methodology and some concise methodology to answer questions in the final exam and also in the written exam. My second point is use the recommended literature. So, I mean, they recommend a lot of books. So, for example, Boron, um, Ganon, and I mean, those books are both very, very good. They're very, very concise. But when I was studying physiology, I was using Guyton and Hall. I used the big Guyton and Hall for some parts, and I also used the Pocket Handbook Companion, which was also good for just having a small or getting a good gist of things before reading the big Guyton or before reading one of the lectures. So I would thoroughly recommend Guyton and Hall. Personally, that's my personal favorite. Um, and then also another really, really key part of the exam, especially with some of the professors, is they like to get you to think about graphs and possible parameter variabilities. So, for example, um, in this particular lecture, we will go through some basic graphs, and you will understand what happens when, for example, you increase oxygen demand or you reduce oxygen demand, etc., and your metabolism demands. So those are very, very important when it comes to getting a good idea of what we can expect the body can do and what we actually uh, see objectively using particular devices, for example, like spirometry or ergometry. And then another really important aspect in learning physiology is also studying biochemistry concomitantly. So, what I mean by that is, I think it's implied by the teachers that you already have a good understanding of some basic biochemistry principles before you do your physiology exam, and it's not uncommon to be asked about some biochemistry in the final exam. For example, I had a renal question when I was doing my physiology exam, and I think one of the professors asked me a lot about biochemistry and the aspects which were relevant to the physiology as well. But I think it's very important that you have a background of biochemistry, or at least a little bit of biochemistry, before you also start your physiology explanations. And then the final thing, which I think is just as important as revising and having all of the information in your brain, is keeping it simple. So KISS or KISS is keep it simple, stupid. Is what I tell myself. And basically, I think it's a very, very important thing um, to keep in mind when you're talking about physiology in particular, because it is quite a daunting task, just because of the volume of information and the actual intensity of the information is quite a lot. So I think practicing composure and staying calm under pressure and being able to think straight, and also being able to kind of back it up and build on the basics. So start from square one and then build it up, build it up, build it up, and kind of add more advanced knowledge onto your already laid foundations. I think that's very, very key, because I think during the exam they will ask you a few abstract things, and they will try and obviously put you off by drawing you out into other conversations about different topics. So it's important that when they do that, you remain calm and composed, and you answer the question to the best of your ability. So yeah, so that's my very, very simple basic study tips and final exam advice. If I could add something to it, I would also go through the MCQs in Ganong, textbook of physiology, because I hear that that's something that the physiology department use, and quite often the variations of the exact questions as well. So I think it'd be quite a wise decision to look through those questions before your multiple choice exam. So the first topic, and really the key topic for integrative physiology, what is regulation and what is adaptation? So regulation is, in the most simplest terms, it's the control of living systems from the cell to the whole organism, and it's an open system. So what I mean to say by an open system is that it depends on external factors and also flows of energy and signaling molecules between the external environment, also the internal environment, and obviously it's in both directions. So it's kind of like a symbiotic relationship with inside the organism and also outside of the organism. And with that, uh, with both of those things kind of locking and keying together, we can get a good idea of the regulation of a particular organism in a system. And if you compare that with adaptation, so adaptation is it's more of a complicated function, you could say. So it's a complex of biochemical and functional and structural changes in a particular organism, and it can be caused by long-lasting or repeated environmental changes. So a really key word that is said a lot of the time when it comes to environmental changes in particular is epigenetics. So epigenetics in this particular term, especially with the term of adaptation, is quite important. And epigenetics plays a very, very important and quite a developing role in how the external environment can actually influence our genetic core. So that's epigenetics, and that's adaptation. And now we're going to discuss adaptation a little bit more. So sorry about my very, very poor handwriting. I kind of wanted to make it more interactive where I could write these things out, but I'm a bit technologically challenged, so I couldn't do that, unfortunately. But so what is adaptation? So an adaptation can be an individual change, or it can also be a genetically fixed change. So individual is something, I mean, it's a very simple thing, because we ourselves can change how we adapt to a situation. Whereas genetically fixed is something possibly you could say it's more related to transmission. So it could be vertical transmission, or it could be horizontal transmission, and especially in today's context with a worldwide pandemic, transmission is something which is very important because viruses are vastly evolving and they're passing on mutations, and adaptation is basically about, or especially genetically fixed adaptations are essentially based on vertical transmission of genetic mutations or potential gene changes that can, or mutations basically, that can better a cell or an organism's function. And what's important about how an adaptation takes place is its mechanism. So it could be a wide variety of things, how it could happen. So it could be that there is changed plasticity of the CNS. So by changed plasticity of the CNS, what I mean to say is that um, our brains, they contain a lot of proteins, and to some extent they are quite plastic in the sense that they can change um, and evolve as time goes on. And they can, essentially, they are plastic, so they can possibly, there could be temporary changes of grey matter. There could be more synapses being formed in order to kind of aid our retention and our memory. For example, we go through this process when we're studying and we're revising, we're trying to create more synapses, so we form more connections inside the brain. And then it can also be, change in organ size. It could be changes in autonomous tone in athletes, for example, especially in the heart, which we'll go through during this lecture. Athletic people tend to have hypertrophic hearts, which is a change from, or which is an adaptation from having a normal functional myocardium. And then also there could be temporary changes of skin color. And what's important is how long an adaptation takes as well. So it could be minutes, or it could be years. It could be generations. So it's always an important factor. So a classification of an adaptation. So classification of adaptations can be separated into two different entities. So we can classify them according to their target parameter. So, for example, we could be adapting to changes in the temperature, or we could be adapting to changes in our diet, or high altitude, or exercise. Or we could adapt according to the output. So this could be at the level of the 5 basic senses. So 5 basic senses are hearing, touching, sight, smell, and taste. So one of those, or something could influence one of those 5 basic senses, and it could also be due to changes of behavior as well. And two more definitions that are quite relevant with adaptations are acclimation, which is a reaction of a whole organism on change in one change factor in an environment, whereas a acclimatization is the reaction of a whole organism on change in several change factors. So those are the key differences. Acclimation is for one change factor, whereas acclimatization is for several in an environment. And our clinical correlation with adaptations is really maladaptations. So, for example, a maladaptation is hypertension. Obviously, you can see in this little picture here what hypertension does. It obviously constricts the lumen, and it reduces the lumen, and it obviously constricts the vessels, increasing the arterial blood pressure, and obviously the resistance as well. And I won't go into too much detail with it as well, as this is a physiology lecture. And then another maladaptation that we as humans have developed is psychosis. So psychosis is, for example, if you're having a psychotic episode, that can be due to excess dopamine. So our bodies have essentially evolved to basically be able to produce too much dopamine in response to some things, and what happens when we produce too much dopamine is that we become psychotic, so we are in a state of psychosis. So these two are clinical correlations with the term maladaptation. And then the next thing we're going to discuss is exercise. So um, we have two different forms of exercise. We can have dynamic exercise. So dynamic exercise, to define that, is basically the movement of your joints. So you're actively moving about and you're actively exercising. Whereas static is you're staying in one position and there's no movement. So an example of a static exercise could be, for example, warming up or doing some stretches. And then obviously we have different types of muscle. So we have skeletal muscle, we have heart muscle, and then we obviously have smooth muscle. And the effect of exercise on our bodies changes a lot of things, and it also influences many different systems, which in classic feedback loop mechanisms try to respond to meet the demands. So, for example, the cardiovascular response to exercise is we increase the cardiac output, right there. So we increase the cardiac output so we can provide more blood flow to all of the necessary muscles that require more oxygen, and also for our lungs. We have an increased coronary perfusion because obviously we're trying to get more blood all around the body and also to the heart, so the heart can keep on pumping and it can maintain a strong chronotropic rate and inotropic rate. And we also tend to have hyperemia in lungs and muscles. So just to define what hyperemia is when you have excess blood in a particular organ or a space. So it does correlate with increased perfusion, and usually you tend to see hyperemia quite nicely in diagnostic imaging and also in autopsies. And yeah, so that's the cardiovascular response. When it comes to the respiratory response, obviously to meet the demands of strenuous exercise, we will need to have a higher gas exchange for aerobic respiration to take place in muscles. So the gas exchange will obviously go up. When that goes up, our diffusion rate will also go up because oxygen is transported to cells, and also there's exchange in the alveoli, and that happens via diffusion. Our ventilation rate will go up because we will be trying to pump out all of the toxic metabolites as well, and also via ventilation we're trying to increase the perfusion because obviously we have a VQ coefficient, which is ventilation and perfusion coefficient, And with increased ventilation, we'll obviously have increased perfusion, so the lungs can carry out the necessary gas exchange in order to meet the demands of exercise. And in terms of metabolism, so we will also have metabolic responses. So these could be changes to the acid-base balance. So what will happen with the acid-base balance is that we will probably have a shift in pH, and it will obviously become more alkaline, and so we will have respiratory alkalosis. And then furthermore to that, in terms of oxygen dissociation curves, when we're talking about the concentration of oxygen needed and the saturation, the curve will be shifted to the left. So when it shifts to the left, we will have a higher saturation at a lower concentration of oxygen. And we will also have glycolysis going on as well in the body, lipid oxidation, amino acids breakdown, and ketone metabolism. So yeah, and in the next few slides we'll kind of go through all of these topics. So in just specifically this figure here, we're just talking about the cardiovascular system. So what will happen when, or what will be the response of the cardiovascular system during exercise. So if we can compare that, the usual cardiac output at rest is between 5 to 6 liters per minute, whereas during exercise, that figure shoots up to 25 and sometimes even up to 35 liters per minute. And obviously, yeah, it's a 4 to 5 times increase. And cardiac reserve. So cardiac reserve is another definition that we need to define. So cardiac reserve is the rate of blood that is passed out of the heart and pumped around versus the amount of blood that could have been pumped out, or the total amount of blood that could have been pumped out across the body. So that's the cardiac output. Obviously, we will have tachycardia, so our heart rate will obviously increase, and our stroke volume will also increase. Our systolic blood pressure will increase, but there's no particular figure that we can be sure that is the complete level of, or that is the final set systolic blood pressure during exercise. Diastolic pressure will fluctuate. It can go down, it can go up, and it can stay the same sometimes, so it's also hard to predict. And our muscle perfusion, that will obviously go up because our muscles will be needing nutrients to aerobically respire. So yeah, and also to create ATP. So this was the heart, and the responses that undergo, or what happens during exercise, really. And these figures and this table here is to describe what happens in terms of the respiratory response according to exercise. So usually minute ventilation at rest is between 6 to 12 liters per minute. During exercise, that figure jumps up to between 19 and 120 liters per minute. Our respiratory frequency is obviously rising, so we become tachypneic. Our tidal volume will obviously increase as well. And the blood flow per liter minute also increases significantly. And then obviously you can see here that if our VO2 intake is increasing, it's only natural that the total carbon dioxide um, per minute is also increasing as a byproduct of respiration. And the PO2, so the partial pressure of the oxygen, the TOR, will be decreasing in order for us to hyperventilate as a adaptation to exercise. And the the oxygen extraction will also be relatively increased. Oops. So this figure here, this is a study. It's quite a common study, which I think many departments have conducted. So on our x-axis we have O2 consumption, and on our y-axis we have total ventilation. So up until the point of moderate exercise, we're roughly using less than 2.5 liters of oxygen per minute. And if we extrapolated this, we would roughly have about 60 liters per minute of ventilation during moderate exercise. But as the severity and the intensity of the exercise increases, this line of best fit, which is taking every single result into account and is sort of the mean, or it's showing the mean values of total ventilation versus oxygen consumption, shows that as the severity of the exercise increases, so the need for the body's the need for oxygen consumption in the body increases, our total ventilation rate and also the liters per minute will also increase. So this, I think, is a particularly important graph when it comes to understanding how a change in oxygen consumption changes our total ventilation. So yeah, and then in terms of actual, this is where the biochemistry comes in particularly useful. Obviously, this isn't a biochemistry lecture, but I think it is important to mention a few points. So ATP will be needed to be produced continuously during aerobic respiration and metabolism, and during short bursts of high intensity exercise, the rate of ATP that is produced and the rate that is required is clearly not enough. So the body's response to creating enough ATP is undergoing glycolysis, and byproducts of glycolysis will be ATP, which we obviously need to carry out these functions, and also lactate and also H plus ions. So I think this is a very key and very important cycle that I think we all probably need to remember. I could go into it, but like I said, this is a physiology lecture, and I think these are technicalities which I think you guys probably know. So I'm probably going to leave this right now. So we're going to move on. Yeah, so like I said, in terms of ATP consumption, the demand for ATP grows and grows whilst we exercise, and aerobic metabolism that does not produce enough ATP for the body's demands during exercise. So what we can do is, or not what we can do, what does happen is once ATP or once aerobic metabolism is shifted to anaerobic metabolism, the anaerobic pathway can produce up to 2.5 times faster ATP. But the downside to producing ATP faster than the aerobic pathway is that it really only lasts for 1 minute of exercise, whereas aerobic can last for hours. And then also another way of producing ATP is through phosphocreatine. So phosphocreatine will also produce will produce a very, very high molar concentration of ATP, but once again it's very short sustained, and it's not really for long-term exercise or exercise lasting a long time. And during low intensity exercise, we tend to get more energy and more what's the term I'm looking for? We get more energy essentially from fats rather than glucose. So as you can see during this graph, at a where x on this axis is exercise intensity, at rest we're using up 60% of fat substrates, which is more than glucose here at low intensity. And obviously, as our intensity increases, the need for glucose increases, and our fat consumption to get energy decreases. So this is also another interesting figure and an interesting graph for us to understand the energy substrate use during exercise. Another one, well, this graph is based on oxygen consumption. So oxygen supply to exercising cells lags behind energy use. So this creates a oxygen deficit. And how do we make up for this oxygen deficit? So we have excess post-exercise oxygen consumption, which makes up for the loss of the oxygen consumption. And this is also quite a interesting graph once again. And I think this is also quite an important graph for your final exam, and I think this is something that you do get asked. And essentially the deficit is compensated by excess post-exercise oxygen consumption. This is somewhat important, but I think the previous graphs maybe have a bit more importance than this, but this is very, very important clinically when it comes to ADGs. So obviously on this you can see that there's the gas partial pressure on this axis here, and then on the right side you've got the total pulmonary ventilation, and naturally during exercise your ventilation rate will increase and increase. However, your arterial PO2 will remain relatively steady and stays the same, whereas your arterial P partial pressure for carbon dioxide actually decreases after a while, and the venous partial oxygenation pressure will continue on decreasing. It doesn't quite reach zero. So we've discussed all of the cardiovascular response, the respiratory response, and also the metabolic response to exercise. But some of the body's adaptations that have happened over increased exercise, for example, in athletes, and also just physiologically, there have been a few changes as well. And so, athlete's heart is hypertrophy. So, in the following slide, you will see that. I mean, this illustration doesn't really show the actual comparison of a healthy heart and a hypertrophic heart. But what will happen is a hypertrophic heart will have an increased myocardial density, whereas a healthy heart will not, obviously. So it will also undergo dilatation, and when a hypertrophic heart undergoes dilatation, the volume reserve will increase by 1.5 times. So why that's useful for athletes is that they can pump more blood around the body in one contraction. But to help them furthermore to that increased volume reserve, they will also have an increased chronotropic response. So they have a larger response. They have a larger volume reserve. So the actual volume is increased, and then the number of heartbeats per minute is also increasing. So you can begin to understand why the demands that their body puts on their body, they can quite easily meet them with this adaptation. Whereas for the mere mortals who are not athletes, we can also undergo physiological hypertrophy, but not to the same extent as athletes. So some of the adaptations that we've developed are prolongation of the muscle fibers. So you increase the muscle fiber length and you increase the thickness as well. And what this does is it can and it may not, but it can. I mean, in most cases, it does increase the contractility of the muscle fiber, so you will get a stronger contraction, whilst also having a possible increase in the speed of ATP hydrolysis, which would obviously be really beneficial for aerobic metabolism. And another key adaptation that's happened in our muscles is that we've developed an increased number of mitochondria. Mitochondria is obviously the powerhouse of the cell. And we've also increased, or what happens is there's an increase in the activity of oxidative metabolism enzymes. So that was exercise and the regulation of the adaptation to exercise. Like I said, athletes will undergo quite substantial hypertrophy, and without going into too much detail, that is known as a cardiomyopathy. You will learn more about that in third year, and as you go along. And cardiomyopathies are also some of the leading causes as to why, for example, some footballers they've been playing football and very, very suddenly their heart stops beating, and that's primarily due to this cardiomyopathy of hypertrophic heart. So I think thermoregulation is something that is quite a nice topic, and I think it is very, very easy to understand once you. I think it's very easy actually. I don't think any of you guys will have any problems in understanding it. But our critical core temperature is about 37.1 degrees, and we can have a fluctuation from 37.1 degrees by 1 degrees approximately either side. So it could be 36.1 or it could be 38.1. Technically, if your body temperature is 38.1, you're pushing quite a high fever. But in the following slides, you'll see a temperature of 38.1 may not be enough to physically damage or cause dysfunction to the body. But how does our body regulate its critical core temperature? So it's the body temperature is the balance between heat production and heat loss, and obviously from our schooling days, we know that heat energy or any form of energy cannot be wasted, but it's merely converted into other forms. So it could either be in production, or it could be in kinetic movement, which causes it to radiate. And heat produced is a direct byproduct of metabolism, and like I said, we can increase our heat production by muscle contractions or increasing our autonomous tone by shivering, or physiologically increasing our thyroxine in the long term. And so that's heat production, and then the rate of heat loss is determined by the rate of heat conduction to the skin and the rate of heat conduction from the skin to the surroundings. So it's the rate between how much heat is conducted to the skin and then how much of it is conducted from the skin to the surroundings. And blood flow to the skin from the body core provides a heat transfer, and it obviously it cools it because it's conducting the heat from the center to the periphery. Right, so what happens, what processes occur during heat loss, and how do we have an increased heat loss when, for example, we are in an incredibly hot environment? So heat loss occurs by radiation, conduction, convection, and evaporation. Radiation usually causes loss of heat in the form of infrared rays, whereas conduction heat loss is by direct contact. So, for example, touching objects and transferring the heat from yourself to the contact to the object, possibly. And convection heat loss results from air movement. So this is what we call the cooling effect of the wind, especially on our skins, which cools it and which reduces our heat and cools us down. And then evaporation from the surface of the skin, but that is more relevant at very, very high temperatures rather than common temperatures that we experience. And another important factor in thermoregulation is also the role of sweating, and that's because sweating is innervated by the sympathetic cholinergic fibers in the autonomic nervous system, and when they are stimulated, the rate of precursor solution secretion is increased. Sorry, there should be an arrow there, it's missing, but it is increased. And that's obviously that makes sense, I think, because if you are trying to control your body temperature, of course, you'll naturally start secreting solutions, which will cause cooling effect on the skin, obviously. But the reabsorption of the electrolytes is constant. So in terms of kidney reabsorption, you wouldn't really expect too much change. But what can change sometimes if there is, a large amount of volume in terms of precursor solution secretion, um, is that the reabsorption still will remain constant, but the actual constitution of the secretion will obviously contain more sodium chloride. And so more sodium chloride will be lost in the sweat in comparison to when you don't produce large volumes. And if the sodium chloride content reduces, this is due to the increased aldosterone, because obviously we know aldosterone is um, an important hormone that is produced when the renin, angiotensin, aldosterone system is stimulated, usually when our blood pressure is low. So, yeah, if the sodium chloride content reduces, this is due to increased aldosterone. And this figure just describes roughly which process takes place more often at you could say room temperature and also in higher temperatures. So at or near about room temperature, 21 degrees, it's not quite room temperature, but at 21 degrees we tend to have more commonly almost 3/4 of output energy loss is through radiation and convection, and almost 1/4 is through evaporation, and then the following 3% that make up our output mechanisms are through breathing and excrements. And in higher temperatures, you can obviously see that evaporation takes a much larger, or it makes up a larger amount of these processes, which will cause heat output rather than radiation and convection, because they just will not be efficient enough to cause enough heat loss at the time or during the time needed for the body to be able to cool naturally. And the hypothalamus is also a very, very important player in the regulation of heat loss and heat gain. So obviously we know that the hypothalamus is made up of the anterior part and the posterior part. So the anterior part, or the anterior hypothalamus, is more heat sensitive, whereas the posterior and the septum and the reticular substance of the midbrain is more cold sensitive. And you might be asking how the hypothalamus will receive this information. So it will usually receive this information via two different transmitters. For the anterior hypothalamus, the mediator is noradrenaline, whereas the posterior hypothalamus will have serotonin, or 5-HT, which will mediate its response. So when we have a high body temperature, what will the hypothalamus do? So the hypothalamus will another thing I forgot to mention. The hypothalamus is obviously a key factor in controlling and regulating the autonomic nervous system response. So the body's response to high body temperature is peripheral vasodilation to increase the blood flow to the periphery to conduct heat loss to the surface. We will try, or we will sweat, and sweating is usually initiated by a 1 degree increase in our body temperature, and that's enough to stimulate sweating. And another mechanism is that we will stop shivering. So any mechanism that the body performs which will generate heat will be inhibited in order for us to lower our body temperature. And then on the contrary, if we have a lower body temperature, we can expect to see our vessels undergoing vasoconstriction and pilar erection. Pilar erection is when your hair on the surface of your skin they become erect, and the reason why they become erect is to trap air next to the skin, just enough air to the skin to essentially gain heat and insulate. And obviously, we will, if a high body temperature, we cannot shiver or follow out mechanisms that generate heat. When we have low body temperature, we will. So we will have sympathetic excitation. So we'll have shivering, as well as an increased muscle tone, and our body will also produce excessive thyroxine. So, like I said, with usually your critical core temperature is 37.1 degrees, and even if that figure will either increase by 1 degrees or lower by 1 degrees, usually we won't have detrimental symptoms. However, if your core temperature does drop to, let's say, 28, which is the lower end of the scale, we can undergo muscle failure. So during muscle failure, you will have a lot of proteolysis and a lot of breakdown. So a lot of breakdown may also increase phospholipase C, and a lot of things will undergo apoptosis, and your enzymes will denature, obviously. And then towards the upper end of the scale for temperature, at 44 degrees, critical core temperature, our bodies just cannot cope with the increased temperature, and usually it will lead to death, unfortunately. So, a clinical correlation to thermoregulation. So, our bodies, when there is an abnormal rise in blood temperature, in the body temperature, which is above normal, usually we call that a fever. So, a fever is our abnormality here, and that can be caused by the resetting of the set point for the temperature control. So this could either be due to bacterial endotoxins, could be due to viral toxins, inflammation, or any other pyrogenic product. And when we've got these foreign particles in our body, they will obviously initiate an immune response. So they will activate the leukocytes, macrophages, and also the killer lymphocytes, which will phagocytose these foreign particles. And when there is phagocytosis, the cells tend to release many cytokines, but particularly interleukin 1. And this cytokine tends to cause the fever, and it also induces prostaglandin E2 to be formed, which acts on the hypothalamus to elicit the fever. And for example, when we take paracetamol or when we take aspirin, those drugs tend to work on this pathway of inhibiting prostaglandin E2 to be formed in order to either prevent the fever from happening or reducing the fever. And when the interleukin 1 mechanism resets the set temperature point, our body will be maintained at that higher temperature, and because the temperature has been raised to that particular level, we somehow undergo subjective sensations of being cold. So quite often we start shivering. On the surface of our skins, we can see piloerection. However, once we've reached the set point of the set point body temperature, we no longer have the subjective sensations of being cold, and usually you could say that's when the peak of the fever has been reached, or the peak of the infection, because after that point the pyrogens start being cleared away from the body, and what happens is the set point starts decreasing back to normal, 37.1 degrees, via a number of autonomic nervous system mechanisms. So we will have vasodilation, sweating, and usually this is known as the crisis or the flush stage, and it's usually a good sign because the fever is coming down, and the flush is because of the vasodilation along the skin, which causes a bit of a inflammatory response locally, hence why we have flushing skin, possibly cheeks or skin. So yeah, it's a good sign. So kind of coming up to the last two adaptations and regulations. So in terms of postural changes, so by postural changes we mean when the body's position changes from lying to standing, or vice versa. It's quite common in the elderly, particularly because what happens as you get older, your baroreceptors, the sensitivity tends to reduce, and your carotid sinus, it functions, but it starts to show a little bit of insensitivity, which will cause orthostatic reaction. So what happens when, usually due to gravity, when we talk about the blood pressure, so the blood pressure in all of the vessels below the heart will increase because there's more blood going down because we're standing up, and the blood pressure in all of the vessels above the heart will decrease because obviously blood is being pushed against gravity. So what happens is that when we lie down, so the sudden closure of the venous valves due to the increased blood pressure and the venous pressure, or the increased venous pressure due to continuous blood inflow from arteries, that will cause the veins to fill up increasingly more, and there will also be an increased dilation of the veins. And what this will do is it will cause an increased venous return. So when we have a decreased venous return, our stroke volume will be reduced, our cardiac output will be reduced, and our blood pressure will obviously be reduced. And so we will have the inhibition of the baroreceptors. This is known as the baroreflex. So essentially, we will not have enough blood flowing to keep us standing, and what happens is when we stand up, we fall down or we lose consciousness. And then subsequently, we will have an increased capillary filtration rate, and this will decrease the plasma volume, so it will increase the level of the ADH, and the renin, angiotensin, aldosterone system will be stimulated, and there'll be increased response from that mechanism to counter the postural changes. And on to the final topic now. So reactions to bleeding. So obviously you can see in this top left image seems to be some sort of traumatic event where the patient obviously has lost a lot of blood, and this can be quite an issue because depending on the amount of blood lost, it can be quite harmful to the body's responses, and it also changes the treatment aspect of things as well, because if there's an increased amount of blood lost, then you might find that management of certain patients changes. Whereas if there's small blood loss, then maybe we might try and use some fluids, as you can see in the bottom right, to resus and to increase more, or to increase the intravascular cell component. So, like I've said, the status of the patient and the feedback response depends on the percentage of blood loss, or blood lost really. So if it's 10%, usually there's a slight transient drop in the blood pressure. So nothing too catastrophic, but something which we obviously observe. Whereas if it's between 20 and 30%, the mean blood pressure decreases quite remarkably, and it's usually between 60 to 80 millimeters mercury. And if we increase that once again in the next 10% compartment, or where it is between 30 and 40%, the mean blood pressure can decrease to about to between 50 and 67.5 millimeters per mercury, and usually at that point the MAP, the mean arterial pressure, is quite low, and it's very, very critical. And anything beyond that point, so 40% plus, we have big issues, and it usually tends to lead to death, because it's just very, very difficult to kind of resusc the body and get enough fluids and control the hemorrhage in the body. And one of the leading, well, not leading, but what happens when we have bleeding? If there's enough bleeding, what can happen is that we can experience hypovolemic shock. So shock is obviously a term used to describe when we have almost multi-organ dysfunction due to decreased perfusion. And obviously in this case, the decreased perfusion is due to bleeding and hemorrhage internally or externally, and that can cause shock, which is a possible another cause for concern. And so we should also treat the shock. So just finally, what changes will happen when we bleed? So I've tried to break it down into a few, into kind of basic bullet points here, without the bullet points. But so you'll have a decreased blood pressure, obviously because blood loss will cause blood to be lost. So your intravascular volume will be reduced. This will cause a decrease in stroke volume. Your cardiac output will be reduced. And so what happens is, in terms of a response for us to counter these three changes, our body will become tachycardic, so we'll have more, that'll be a chronotropic response, and your respiratory rate will also increase in order to create enough exchange for whatever blood vessels that need nutrients for them to get it. And that will cause tachypnea. We'll obviously have reduced perfusion because of the reduced cardiac output, and it will also be due to vasoconstriction, which will cause an increase in peripheral resistance. And if you've got reduced blood leaving the heart anyway, and there's more peripheral resistance, your perfusion will be reduced, and so anything, any blood leaving the heart during bleeding has a lot of resistance to overcome, and it will obviously cause a decrease in renal perfusion. So renal perfusion, the kidneys are incredibly important. And if we have less blood flow to the kidneys, it means that less urine is produced because less blood is being passed through the glomerulus and is being filtered. So we'll have less things being excreted, so less waste and harmful toxic substances being excreted, and so this will cause uremia, and it will be increased uremia. So uremia is anything toxic and also nitrogenous waste that will accumulate in the body, and potentially that could be quite dangerous, and once again it can cause a critical situation, and our kidneys will respond to the low blood pressure and the reduced cardiac output by activating the renin angiotensin aldosterone system, and we will also have an increased amount of ADH being secreted to reabsorb the water from the kidneys to increase the intravascular volume. So those are the instant changes, and then what happens maybe within hours or between hours and days, a bit later on, if the patient survives, is that we will have a decreased capillary hydrostatic pressure. So hydrostatic pressure is the pressure inside the vessel, which is pushing it to the interstitial compartment. If that's reduced, that basically means that the pressure, because they're in equilibrium, so your oncotic pressure and your hydrostatic pressure from inside the vessel is at equilibrium, which enables, you know, for there to be no kind of transfer, no edema, etc. And if there's too much bleeding, then naturally we don't have enough volume inside the vessel, so the hydrostatic pressure will decrease. And because there's more pressure on the outside, so there's actually reabsorption of fluids inside the vessel from the interstitium. And what this can do is it can cause an increased volume in the intravascular component, and it can also possibly cause fluid overload, which is another big red flag, because this could cause a number of failures. This could cause heart failure, and once you kind of reach that state of acute heart failure, especially if there's a lot of bleeding, that's a big red flag, and unfortunately the prognosis is quite poor. So that's what can happen after the instant short-term changes. You're talking maybe a few hours after. So that's what can happen. So that is the final point for reactions to bleeding. And I'm now coming up to the I finished my lecture now, and so really it's up to anyone watching now if they have any questions. So if they have any questions, they can just write on this chat. So if you want to do that now, you can type away now, and I'll try and answer them right now. If not, or if you still want to write, I just want to explain. My sources for this lecture were Guyton and Hall, textbook of medical physiology. I also used a lot of graphs from Silverthorne Human Physiology, and to kind of finish off things, I used lectures from Professor Beberova, because she's a professor who particularly likes graphs and asks about them in extensive detail. So I can't promise you that all of the graphs that I've used will come up, and I can't promise that I've used all of the graphs as well. There's so many graphs out there that it's hard to kind of know what could be useful and what may not be useful, but I think I've added the ones that are useful, so hopefully they will be of use. But yeah, I can't exactly hit all the points sometimes. And yeah, so if there are any questions, you can message me on this chat here, or if not, you can message me on Facebook or on Instagram. Very easy to find on all of those platforms. And yeah, I mean, thank you for listening. If you listened, thank you for tuning in, and I hope it was useful. And yeah, thank you for your attention. And just before I kind of shut things down, if all of you could, if anyone watching has free time, if they could just complete this feedback form, it would be much appreciated, just for us to get a rough idea of what to change or how to make things even better next time. But yeah, thank you for your attention, and I hope you all have a nice weekend, and thank you for tuning in, and stay safe.